Can you hear me, guys? Yeah. All right. How are you? How was the break? Not bad. Any parties? I have like 3 students or 4 that didn't show up yesterday to the quiz, so I'm guessing so. Yeah. 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 I have to restart the wheel there. So next week it will be on 7. Okay. This week is on 6. What did I want to say? So I have just a few updates for you guys. Midterm should be coming in tomorrow or Thursday. I'm still waiting on IETS to scan those sheets. And yeah, and then we can talk about the questions and all that. I was planning to do it today to go over the questions, but I know that some students have an exam tomorrow, so I'm just not going to do it today. I'll let you go a bit earlier, and then we'll see what we do with that. Okay. What else? The final exam is downtown. We just got the room, so it will be again on Thursday, April 23rd from 9 a.m. to 12 to noon. There's 3 rooms that are assigned. So I guess on your what is it called? Concordia account, you should have the one that's specific to you. Okay. Just be careful with this year. So MBS is 2nd floor down. So it's the JMSB building, the floor of the, of the metro. Okay. And the rooms are somewhat hard to find. I remember these rooms specifically because I had exams here. So go a bit earlier to try to figure out where they are. Okay. ACSD students, you know where you are. You'll get it in your account. Don't get lost. But yeah, that's pretty much it. Do you have any questions for me? No? Okay. I might be a little bit boring today. I'm sleep deprived because I messed up my sleep cycle like 3 days ago. So doesn't want to get fixed, but it's okay. So chapter 7. Now we are getting into the stuff where you have less problem solving and more learning by heart. Okay. We'll get back to the problem solving, but only for chapter 11, I think. And that's related to the lac operon and the gal operon. Okay. Other than that, everything by heart. Okay. This is like a typical multiple choice question. Okay. Just for this group here, do you guys see properly, or should I close the blinds? You're good? Okay. Anyways, we want to see the sun, right? So what did I want to say? So up to now, we've been looking at the development of the field of genetics by looking at genes as being abstract units that are inherited. So we do know from CJEP and all that, but we don't really know what it is specifically, right? What we learned throughout the few lectures ago is that the genes must be faithfully replicated. So this way you can inherit them because you need one copy in the parent, one in the other, in the offspring. It must contain some sort of information coded within it, which allows to determine a certain phenotype. It must be able to change. So this creates the variation, which we know as alleles. And then we know that there's one gene, one polypeptide. So each gene produces a protein or a polypeptide, which then has a certain function as an enzyme or whatever. And that they are carried on chromosomes. And that these chromosomes are made of DNA and proteins. It's important to remember that chromosomes are not just DNA, but they have also proteins on them, which are called scaffold proteins, which keep the chromosome together. Okay? So there are a few experiments that were done following all this knowledge to determine what's at the molecular level, what is a gene kind of, right? And it was done by a few people, and we're going to go over each experiment today. Because I'm talking about experiments, you need to remember the experiments for the exam. Okay, so I can be clear about this. It's important to know who did what, because you'll be asked for the McLeod experiment. They did this or that or that. Okay. So the first thing that led to the discovery of these genes and the understanding that there's actually some sort of molecule that leads to inheritance, it was done by Frederick Griffith, and it was done in bacteria. So the first thing that led to this discovery is the concept of transformation, which we've seen in lecture or chapter 6. That's better. And this was done in 1928, so not that long ago, about 100 years. And what this guy did is he took 2 strains of bacteria, Streptococcus pneumonia. You probably had pneumonia when you were younger or some sort of variant. So this is the same one. And he took 2 different strains. So 2 different strains of these bacteria. One of them is this one here called the S strain, which has a smooth coat on top of it. So you have the cell and it's coated with a smooth layer of polysaccharides. And then you have the R strain, or the rough strain of this bacterium. And this one does not have the polysaccharide, if I'm not wrong, or has a different type of polysaccharide. The S strain, when you inoculate mice with it or any other organism, including humans, it can kill you if not treated. Okay. The R strain does not kill you, probably because it's lacking the virulence gene or something like that, right? So one way to kill the bacteria is just to heat it. When you heat it, the bacteria will die and it will not become virulent anymore. So what this scientist did is that he heated the S strain and then mixed it with the R strain, allowing for transformation to happen. And then he inoculated the mouse with this mixture, and it led to him finding live S strain. So there's some sort of molecule inside these bacterium that is taken up by the non-virulent one, and then it's used to become pathogenic. So then he was wondering which type of molecule is it, and I'll get back to this. But they were wondering what was the molecule that was leading to this pathogenicity. Most people thought it was proteins because they are complex and they have different shapes and forms and can be super big, super small. So it seems like it must be the thing that is inherited between different organisms, because DNA and polysaccharides are quite simple compared to, let's say, DNA. DNA is quite simple compared to proteins and polysaccharides. Okay, and so there's another group, the Avery, McLeod, and McCarty group in 1944, which tried to figure out what type of molecule that was being inherited. And what they did is they did the same thing. So they took these S strains, so the virulent strain of pneumonia, S pneumonia, and they destroyed different components of this bacterium. And they did this by using different enzymes. So you can target a group of molecules, the major macromolecules, by using different enzymes. For example, lipases will degrade lipids, DNases will degrade DNA, proteases, proteins, and then you can have different types, right? Like phosphatases will remove phosphate from, right? So anything that has A's at the end, usually it means that it's an enzyme that acts on a group before, right? So if you have kinase, it means it adds a phosphate group. If you have phosphatase, it means it removes a phosphate group. Okay, and so in this case, we're interested in DNAs, RNAs, protease, lipase, polysaccharides. There's a different way of breaking them down, but it's still an enzyme. Okay, depending on which polysaccharide you're dealing with. And so they decided to inoculate this strain, the mixture of this strain with different enzymes to break different molecules. For example, in this case, they used lipase to destroy the lipids, but they kept everything else intact. And then they mixed this with R strain and looked if the mice died or survived. If it died, that means that it was not the polysaccharides or the lipids, and it's something else, right? If it survived, that means that's the thing that you, so if you destroy the DNA, then you lose this virulence, right? And because you lose this virulence, there's no transformation of DNA happening, and so you lose the virulence, and so the mice survives. And this is how they found that it's DNA that is actually the inherited molecule. Does this make sense? Simple experiment? Okay. So I'm saying here it's controversial because what I just mentioned earlier, the fact that DNA is very simple compared to proteins, lipids, and all that. So then there's another group, Martha Chase and Alfred Hershey, who wanted to determine what's found in viruses and what is the genetic material of viruses. Okay, So they did this experiment where they labeled, so in the virus, there's two things, either sometimes three, but most of the time you have protein, DNA, or RNA. And then some viruses do have the envelope, which is lipids. But in the case of bacteriophages, they don't have an envelope. So you have protein and DNA, that's it. So what they did is that they labeled the phage, so two different populations of phage, one of them with radioactive sulfur, which is only present in proteins, right? And the thiol groups of amino acids. You learned this probably in biochemistry. And then, or they use radioactive phosphorus for DNA because there's a phosphate group in the DNA, right? And they were thinking that when they infected bacteria, whatever material is being transferred will actually emit some radiation, and you can see where it is inside the cell. So they used the T2 phage, which is similar to the T4 we learned, just a different one. And they labeled the proteins with 35S, so sulfur, or another group of phage with 32 phosphorus. And they infected two different populations of bacteria, then they broke them down, etc. Okay. So when they imaged this here, they found that the radio-labeled molecules were outside and not inside the cell, right? So proteins were not going inside the cell. Then when they looked at the other group where they used 32 phosphorus, they found that the radio-labeled molecules were actually inside the cell. And so now you know that the phage actually transfers DNA because of this experiment, and that it's found here in the head. Okay? Simple? Easy? Cool. Wait. Okay. So let's dive in a little bit into the building blocks of this DNA. So DNA is super simple. It has 4 different kind of molecules divided into 2 groups. Okay. So the components of this DNA molecule comes from these here. You have purines and pyrimidines. Purines have a dicyclic nitrogen base, while pyrimidines have only one. So what I used to use as a trick to remember this for the exam, pyrimidine is the long one, so then the opposite, so it's the opposite of it, so it's a small one, right? While purine is very short in terms of letters, so it's the dicyclic. You see what I mean? Just the opposite kind, okay? Long one, less circles, small one, more circles, okay? So the molecules are made of 3 components. There's the phosphate group, there's the sugar, the deoxyribose sugar, and then there's the nitrogen base. So let's start by the phosphate. The phosphate here, you could have 3 different phosphates attached to this CH group, okay? But in the case of DNA, it's only a 1 phosphate group, and then you have a 5 carbon sugar, or you call it, it's a pentose, but you call it ribose, okay? And on the second carbon, so if you start counting carbons on this molecule from this one here, so the 1 is where the nitrogen base binds. And I'm mentioning this, it's important, okay? Which carbon is being bound by what, and etc. Okay, it's important for this class. Probably in biochemistry, you'll have to probably redraw this whole thing, etc. Okay, so the first carbon here of the sugar is where the nitrogen base binds. The second carbon, you have an H, and if you had, so it's an H, that's why we call it deoxy, because there's a carboxyl group missing. If there was an OH here, then you would call it ribose instead of deoxyribose. And we'll see what kind of molecule is ribose. Okay, it's RNA, right? So for DNA, it's deoxyribose. It's at the 2' carbon. And then at the 5' carbon, you have a phosphate group. Okay, so that's the first thing. Second thing, so there's 3 components, and then each carbon has an important role. The most important ones are the 1, 2, 3, and 5, and we'll see why the 5 in a second, okay? So these nucleotides have a nitrogen base, as I said. There's 4 types of them: the adenine, guanine, cytosine, and thymine, okay? Not to mix you up, but probably in your studying, you're going to get mixed up with this. Cytosine sometimes will make you think of cysteine, and these are two different things, okay? And you'll need to be able to differentiate both, because cysteine is an amino acid, and we'll learn it in chapter 9. But for DNA, it's cytosine. Thymine, the others are okay. Does this make sense? Okay, so this nucleotide here at the top is called the deoxyadenosine 5-monophosphate. Okay, these names are important. If you've ever took bio, probably you did. If you know what ATP is, right? So ATP is your energy block. Well, ATP comes from this here, deoxyadenosine 5-triphosphate, right? And then the ATP is used with two reactions that remove the phosphate to turn it into the AMP. Make sense? A little bit of chemistry for tomorrow. What else? So that's pretty much it for this, I think. Did I forget something? No. Okay. Purines, small, dicyclic, and then pyrimidine. How do you call it? This when you have two circles instead of one. Not dicyclic, but there's another one. Sorry? Bicyclic, no? Bicyclic. I think there was another word, but anyways, you'll figure out tomorrow in the exam. Okay. Right. So there's an interesting property about these nucleotides that you have to remember. That you have to remember, and it's coming from Chargaff's rule. So this guy called Erwin Chargaff, he found that there was a pattern of in the composition of the DNA. Okay, and he was looking at different organisms, as you can see in the table here, and he was looking at the components of this DNA molecule, which is just a chain of A, G, C, T, whatever, in different orders. And he saw that for different organisms, there's different patterns of these nucleotides. But one thing that was clear, and he made some rules from it. Now, obviously, let me first say the rules, and then I'll tell you the issue I have with this here. So the first rule is that the amount of A, the percentage of A is the same as the percentage of T. That's what he realized. The second thing he realized is that the percentage of C is equal to the percentage of G. So this is adenine, thymine, cytosine, and guanine. Okay. So basically A plus C equals T plus G. And then the GC content is different in different organisms. Okay, so the amount of G and C. Make sense? So if you look at this table here, you will see that it's not really true. Okay, technically, and we'll see it in a second, but technically you should have the same amount of A and T because they interact together. So if they interact together, you need to have on opposite sides of the DNA molecule the same amount. But you'll see that it's not really the case, right? For example, if we look at E. coli, they have 26% of A and about 24% of T. Okay? The reason why this happens is that sometimes they still do bind to the other group, so the G or the C. And this is where mutations happen. Okay? This is the basis of mutations. When there's a mutation, usually what happens is that an adenine will be changed for guanine, and then it will suddenly bind with a thymine, which does not make sense. I'll show you why. And so then the replication of this leads to different proteins or mutant proteins. This is why you don't see exactly the same amount of A and T and G and C. Yeah. Exactly. So the rules that were made at that time were kind of, the 3-to-1 ratio is the same thing, right? It's close to 3-to-1, but it's more like 2.85 to, whatever. Okay? It's the same thing for the organisms. And then what's interesting, without getting too much into this, but the more GC you have, the more resistant you are to heat. And so now you can imagine different microorganisms that live, let's say, in very hot places, right? So the GC content can tell you a lot about an organism, and these patterns are specific for different organisms. Okay, a lot of evolutionary biologists like to study this. In addition to that, how important this is when you're making your own plasmid or when you're copying DNA molecules, you need to be careful with the GC content because it affects the efficiency at which a plasmid is basically copied. Okay, without getting too much into detail. So when we're building a plasmid, we have to be careful with these numbers here and play with the sequence until we get to a specific number. Because if you have too much GC, for example, it will take more heat to open the DNA to separate it, and so you can actually destroy your molecule from there. Okay. Some organisms are able to survive, like archaea. They're usually richer in GC content. Okay. First question. Very simple, but if there's 20% of T in the DNA, double strand, what is the percentage of G? I know you have the answer. 30? Yep. So it is 30 because you have 20% T, so that means you also have 20% A, technically. And so what's left is 60. 60 divided by 2 because you need half is GC, half is G, half is C. So that gives you 30. Make sense? Cool. Okay, let's go back to the other question. We should have maybe... Okay, this question here, I put it here just for you guys to understand the question style because maybe... I'll just say I was unclear, okay? So I'm putting this here to remind you to study the history, okay? In this case, okay? So you would have probably questions like this. Why would Griffiths decide to mix up the non-virulent and dead cell together? So if you don't remember this here, probably you're not going to be answering this, okay? So try it. I don't like, yeah. No. You know why the profs put the dates here? So that they remember them when they're teaching. But no, you don't need to remember the dates. It's just these are key experiments. If you're gonna end up biologist one day, so it's important to know them. Okay. So, anyways, not just waste too much time on this, but it's just to determine if there's some sort of molecule, non-living material that could establish virulence. Okay, that's the answer. Okay, so we have the components, but how did we get to the structure of the DNA? And we talked about this in the first lecture, I think, or second, and I'll tell you why we talked about this. But there were three pieces of the main puzzle of DNA that were used to actually determine the structure. Okay, the first thing is that DNA is composed of 4 different nucleotides, just like we saw. The second one is that the percentage of A and T is the same, and the percentage of C and G is the same, so suggesting that there's actual pairing. Obviously, this is not perfect, but it's close to it, okay? And then there's this lady, Rosalind Franklin, that did an experiment using X-ray diffraction, I don't know how to say it, but, and she found that the DNA is helical, like a staircase, the emergency staircase behind your house, if you do have one. And so her experiment is basically just adding radio-labeled isotopes to the molecule and then just throwing X-ray to it, and then it would emit back a specific light. And it looks like this, right? Then our buddy Watson, that we saw at the beginning. This is the guy that I told you that was saying that different races have different levels of intelligence, and he was trying to base it on science. So he got the Nobel Prize for discovering the structure, and then he lost everything. But him and his buddy Crick, what they did, and these guys super young and discovering shit. What did I want to say? So these two guys actually found out the structure of the DNA by using crystallography in part, and they found that the DNA is like a helix or a double helix, okay? And it has 2 strands that interact between each other, and these strands, yep. Oh, yep. Okay, so you guys know, no. Well, that's the thing, yeah. Well, clearly, everyone did everything, and then they just came like, oh, this is the molecule. But people are saying that they did steal it from Rosalind and then published it under their name. Which, karma comes back. well, this guy is, I mean, he has his career and all that, but this is what you get for stealing. You start saying random stuff like race affects intelligence, and then you lose your whole career. So no point into this, right? But yeah, there's a lot of stealing in science. How does it happen? Usually it's not like stealing, just like come and I steal your data. It's more like you would do an experiment and discover something small, and then I would take that and use it and build something out of it, and then you don't get the credit for it, right? And it happened through many occasions because it's very hard to delimit when was something discovered or not. Because I could have discovered it 10 years ago, but because you were faster at publishing, you just took a little part, right? Then you would get all the credit for it, even though I found it before you, right? So it's a bit complicated like this. Actually, in some conferences, there's people that take pictures of the posters. So you're not, if you go to conferences, you're not allowed to have your phone out, which is kind of weird, but it's because people take pictures of the posters and then just go and they have a huge team and they just do that research, publish the paper and scoop you, right? And the reason why there's this kind of vibe inside the research community, it's because if you have your own lab, you need to publish to get grants. You don't publish, you don't get grants. And so brings you to kind of a mindset where you're fixing data, using Photoshop, how you should not be using it. And as I showed you earlier in the semester, the president of Stanford got into a lot of problems because of this kind of behavior. And no one is immune to this. If the president of Stanford did it, you can imagine the whole freaking down there, especially in students who need to, defend their thesis or whatever, right? So yeah, there's a lot of stealing, but this one here seems more of the type of like taking it from Rosalind. I don't want to get into the politics of this, but c'est la vie. Anyways, let's get back to this. So they all found, okay, when I say they, I mean everyone that contributed, okay, not just these two guys, but they found that there's a double helix with two anti-parallel strands. And I'll tell you what anti-parallel means, but basically they are intertwined like this, and they have nucleotides that bind to each other in the middle, right? The blue strand here is just a sugar phosphate backbone, meaning that you have a sugar molecule bound to a phosphate, to a sugar, to a phosphate, etc. Okay. And then if we look at the 2D structure, it looks pretty much like this here. So you would have on the left, let's say the 5' to 3' strand, and this is where 5' to 3' is important, and you'll need it throughout your career. So focus, okay? 5' to 3' just means that the phosphate group is attached to the 5' carbon of the sugar molecule, which then that sugar molecule is bound to another phosphate at its 3' carbon, okay? And then this next phosphate is bound to the 5', 3', etc. Does that make sense? Okay. It's important to remember this because then you will be doing transcription, and transcription follows a specific pattern, 5 to 3, sometimes 3 to 5, and then depending on which strand we're talking about. So it's important that you understand what's happening here, because RNA polymerase cannot bind anywhere, which we'll see later on, but it cannot bind anywhere. So if I give you a 3' to 5', don't start from that side, okay? And I don't want to mix you up, but we'll get there in like 2 or 3 lectures. On the other side, because they are anti-parallel, so then you have a 3' to 5' strand. So they're just flipped like this, and then it's basically the same thing. If you start from the bottom here, you have a phosphate that's bound to a 5' carbon of the sugar right here, which is bound at its 3' carbon to another phosphate, etc. Cool. In the middle, you have these bases binding together. And these bases, most in theory, A binds to T, adenine binds to thymine, because there's only two groups that can interact and make this hydrogen bond here, right? While cytosine and thymine, they have a triple hydrogen bond because they have 3 groups that can interact between each other. So this conformation here, this conformation here is much more favored than having the A binding with the G or the C with the T. It does happen, and that's the whole basis of mutation. But in theory, this is what should be happening. Cool? Easy? Okay. Perfect. So how did these guys figure out the structure? At least, yeah. So they used all the information gathered by other researchers and probably stole from them. And they looked at the crystallography, not the crystallography, but the X-ray picture that I showed you earlier, right? And then they tried to make 3 different hypotheses to figure out how it was structured, right? So they were thinking, this is the diameter of the double helix by just looking at both sides, right? And then if you had 2 pyrimidines, then the DNA would have been too skinny. If you have 2 purines, it would be too wide. But if you had the purine and a pyrimidine, it came perfectly aligned with the structure. And then you have obviously the hydrogen bonds in the middle, right? Obviously, again, this is not perfect. Probably this thing was a little bit off here and a little bit off there, but at least it's very close to the model they're proposing. So this is how they found that it looked like this. By looking at the size of the different nucleotides and trying to figure out if they would fit in the diameter determined from here. So if a question asks you in an exam, for example, what information led to them figuring out the structure, was these 3 things here. You need a pattern, which I just told you, which you figure out the diameter from. A and T, you need to know that A and T bind together and C and G bind together to be able to do this thing here, etc. Okay? Ça fait du sens? Good. Not bad. Okay. What else? Okay, now let's get into the bonds a little bit. So there's 2 different bonds that you need to remember here. The first one is the hydrogen bond. So the hydrogen bond occurs here between the 2 nucleotides, right? This is a weak bond. You probably know all this, but you gotta repeat. So this bond here is very weak. So if you heat it, it will dissociate very simply. And then you have the phosphodiester bond, phosphate group with the sugar diester. Okay. Between the phosphate and let's look here, between the phosphate and the sugar. So it's called a phosphodiester bond or linkage. And this is a covalent bond, meaning that it's a bit harder to break. So this is one of the interesting properties about DNA, is the fact that you can just heat it a little bit and then it will open up. And you can then copy it or whatever. Okay. And we use this a lot in the lab. For example, if I want to copy a certain gene, what I do is I heat it up, my mixture a little bit, so then the DNA opens, and then I add a polymerase, and it starts recopying. And when I want to close it, I just cool it down, and then it closes back again. Okay, so that's one thing. What else? Sugar phosphate backbone, I said that. 5-3, said that. And okay, any questions? Going too fast? No. Okay, a little bit more about the double helix structure. So the double helix structure has some two different types of grooves, and that's just based on the formation of these pairs here. So there's a minor groove and a major groove, and it's just linked to the size between this and here and here, and between here and here. Okay, all I want you to remember about the grooves. What else? And then this ball model here is something we use a lot of in chemistry. It's just to look at the 3D structure. Basically, it looks like this. Nothing too crazy about this. What else? Crazy from out. Okay, settings. Yep. Oh, yep. Just that there's a minor one and a major one. That's it. Yeah. Very, very simple. It's actually quite useless as information. I don't know why we keep teaching it, but there's nothing we do with major groove and minor groove. Maybe the crystallography people there in chemistry, but not the biologists. Okay. So I put this here. You don't need to remember it for this course. Write that down. But you'll probably need to for biochemistry and all the other classes. Okay. If I remember correctly, we were learning all the different like molecules in glycolysis and where the carbons went and all that. So this is something you would do probably in biochemistry. Okay. This is just to show that there's hydrogen bonds that bind that interact between the two nucleotides. If it's a cytosine and guanine, it's 3. If it's a thymine and adenine, it's 2. And that's it. In this case, I don't care about the numbers. Okay. Yeah. Well, you need to know that purines are these two here, are this one here and this one here, and pyrimidines is the pyrimidine is this, and purine is this and this. No. Yeah, not it. It's useless. But no. Even the carbons and all that, the nitrogens and all that, it's not important. The reason why I'm focusing on the carbons and which carbon it is, it's because the directionality is important. The 5 to 3 prime will be important later on. But knowing that this nitrogen here, nitrogen number 1 has an interaction with 3, that's kind of useless. Good. Okay, so the discovery of this structure was very important because this is what led to understanding that it encoded information. If you remember the slide from Dylan where he had like just a lot of letters on the board, well, that's basically your DNA, and it's composed only of these 4 molecules. Okay, we will see in chapter 9 why only 4 different molecules are able to make the code for 20,000 genes. Okay. And what else? And this structure here was very important because it allowed to understand the replication mechanism and how replication in the DNA happens. Okay. What did I write right here? So this is what they wrote just about they were already thinking about a copying mechanism because of this structure. Obviously, their research is a bit iffy. Some people think that they stole it, etc. But if you want to read the original paper, just write Watson and Crick, Nature, which is the top publication in the field, 1953, and you'll find the paper. Okay. It's important if you are going to go into research in the future to kind of get acquainted with these papers so you can kind of become knowledgeable of what was done in the past, but not really necessary. You'll see that there's a huge difference between before and now. Before you had just to find the structure of the thing. Now to publish a paper, you need like 10 pages of data. Some cool stuff. Okay, the last thing for today. So I just want to mention this before I let you go. So I'm just gonna show you the experiments that were done to figure out the mechanism of replication of the DNA. Next lecture, we'll start delving into the replosome and all that, and all the proteins involved in this process and how they interact with the DNA. Okay, but there were a few hypotheses for the DNA replication. And Watson and Crick were the ones to hypothesize that it was semi-conservative, because if you have a molecule of DNA like this, and then you have two strands, then you can have different modes of replication. You can have the semi-conservative, which it would open up, and then you would end up with so the parental strand with the copied strand, or you can have conservative, which the two parental remain together, and then you would have the two that are copied together. So this is conservative because it conserves the initial form, or the dispersive, which is a mix of both. Okay. So the experiments that were done to figure this out were done by Messelson and Stahl, and they did this using E. coli. So remember that these have nitrogens in them. So what these two people did is that they took E. coli and they grew it in the presence of a heavy isotope of nitrogen, the 15 nitrogen, and they produced a DNA of high density. Then they removed the media and replaced it with media that contained the light isotope, so the 14N, okay, for 1 cycle or 2 cycles of replication. And then they use this gradient of a chemical called cesium chloride to be able to separate the different strands in the tube, okay? Anybody, do you guys know what ultracentrifuge is? Okay. So it's just a machine where you put tubes in it and it rotates very fast and it basically sediments anything, okay? So if you have, for example, let's say a blood sample, you're able to put it in this machine. It will rotate super fast and then it will separate, let's say, white blood cells from the red blood cells itself, okay? Does not happen just like that. Usually you need to put a gradient. So this is why we're using a gradient. So what happens is that based on the weight of this, let's say a cell or a protein or whatever, it will get separated, okay? And the gradient just means that you have this molecule at different concentrations along the tube. So let's say from 2% concentrated to 50 or whatever, okay? These ultracentrifuges are super fast. We have some here in the SP building. They go to 100,000 Gs. So 100,000 Gs, if you're going to the space, the International Space, station, you're usually put at like 65 Gs. And it's like the top of the top train. So 100,000 Gs, you basically implode on yourself. Okay? Some crazy stuff there. So in the labs, if you ever end up in the labs, you'll probably see students not balancing it out because you have the torque happening. So if you don't balance it out, and if it's an old centrifuge, it will start moving by itself. Just like your washing machine, same thing, but just faster, okay? So be careful with this, okay? And ultracentrifuge, they have systems to detect the weight. So it does not start because otherwise it will destroy the wall. Okay, so they did this. They extracted the DNA. So you grow these bacteria with 15N, wash it off, and then allow them to replicate, wash it off, and then put the 14N, let it replicate once or twice, and then you put it into this tube with this gradient, which will separate different molecules based on their weight. They will travel through the gradient by being pulled from the centrifuge force. Okay. In the case where you have this model here, wait, which model is this? It's right here. In the case where you have the semi-conservative model, okay, the parental strands, when you allow it to replicate into the 15N, you will end up with one strand that is 14N and one strand that is 15N, okay? So it will create a hybrid that will be between, in the gradient, that will be between the heavy and the light DNA molecules. And so you grow it into 15N, which leads to this, because the regular isotope that is mostly found in nature is the 14N, not the 15N. So DNA usually when it replicates into 14N. It integrates the 14N into its molecules. Okay. So when you grow it into the 15N, you end up with this hybrid DNA molecule, because you would have the parental. Am I flipping this? Anyways, it doesn't really matter, but basically you will end up with the in this case it's the parental is 15 because he started with 15, okay? But it doesn't really matter. The idea here is that if it's semi-conservative, you'll end up with one that is 15N and one that is 14N, because the 15N will be the original one and the 14N will be the one that's added, right? Does that make sense? It's a bit all over the place, but yeah. So then when you run it inside this gradient, you'll end up with a hybrid DNA molecule that comes at the middle here, okay? Then if you allow this to run again, and I think it's in 14N, yeah. If you grow it again in 14N, you will end up with these two molecules separating, and you'll have a blue strand at the top and yellow strand here. So the blue strand will still end up being replicated with a yellow strand, because the yellow is the 14N, right? And then the yellow one will be also replicated, but it will be replicated to a 14N, because there's only that 14 nitrogen that is integrated. And so in this case, you'll end up with one light strand because it has these two, and then a hybrid one in the middle. Does this make sense? Very good. Okay. Yep. Exactly. So when you're replicating DNA, so let's say you have a DNA molecule that is only built with these things, the purines and pyrimidines that contain only the 15N isotope. So you'd end up with two strands that are heavy. Then if you wash the 15N isotope from the media so that it cannot use it to build the other strands, and you add instead a 14N isotope, then when the replication happens, the new strand will be built only with the 14N, right? So then when they come back together, you'll end up with one that is 15N, one strand that is built only with 15N, and then the other with 14N. Does that make sense? Well, a little bit more specific. Oh no, we don't want this. This is just the experiment that was used to determine what type of replication mechanism happens in reality. Is it the semi-conservative, conservative, or dispersive? Yeah, it's just to differentiate between these. Okay? Right. So that's pretty much it. If we were to do it with the conservative replication, you would end up with two different strands, right? Two different molecules, either the heavy one, which is this one here, or the light one. So if you were to look at the tube, you would have one at the top and one at the bottom, nothing in the middle. Make sense? Okay. Dispersive replication. This one here, you would just have a kind of a gradient inside the tube because you'd have different size molecules. And so because you have all of them stacked up, then you'll end up with a whole tube filled with different levels. Okay? So it's not the dispersive for sure. It's the semi-conservative. Okay, question. Can you guys try to answer this? So they're saying that these two experiments where bacteria was growing in heavy nitrogen for one generation and light nitrogen for two, predicted that the third generation conservative replication would show. What would you find if you did the cesium chloride experiment? If it was conservative replication. Sorry? A medium heavy and a light band. Yep. Make sense? Why? Yep. Wait, 1 second. Let me reflect here. For one, so you start with a 15. Yep. No, my slide is more than me. Okay, here's the answer. Okay, where was I? Wait, let me just think before I. Yeah. Did I make a mistake? Medium, heavy, and light band. So first generation is with 15, and then it splits up like this. No, it split. This is why. I think this is unclear. Okay, this is unclear. What I really meant here is that the first two were semi-conservative, and the third one is conservative. Okay. Sorry about that. If you spot stuff like this, tell me, because in my head they're clear, but then you guys don't see what I see. Okay, so I might make this mistake. But in their experiment, what happened is that they found that it was semi-conservative, and my idea was just to change the third generation to conservative to see if you're able to follow that. Okay, so if the second generation looks like this, and then you go to the third generation, you'll end up only with a light chain and a medium chain because it's conservative. Okay, is that clear? Okay, and that's it. Yeah. Sorry for that. Any questions, guys? No? Are you ready for tomorrow? Kind of. I think they have an exam, organic chemistry. Yeah. What's tomorrow? All right, guys. I'll see you Thursday. Okay.